abbreviated as GMOs. So what I'm going to do is I'm going to tell you what are GMOs, how they are made, how they are tested, how do we test for GMOs, and what are the regulations in our country, and also some of the crops which are GMOs. I'm not going to talk on whether it is correct to have GMOs or wrong. So this webinar is basically a science-based webinar on genetically modified organisms. So, what is a genetically modified organism? So, it involves actually the insertion of DNA from one organism into another, or modifying the organism's DNA in order to achieve a desired trait. So, what it is you are fiddling with the basic DNA to get what you want. So, what you see on the right hand side is mice expecting the expressing the green fluorescent protein, and here you have a blue rose which has a different color altogether. So GMO is genetic modification where you can insert a DNA from another organism into the organism of your choice to get the desired trait that you want. So the definition for GMO is it describes GMOs are also known as LMOs, living modified organisms. They mean exactly the same. This describes plants, animals, and microorganisms which have genes introduced them by means other than by the combination of an egg and sperm. That is, other than by fertilization or by natural bacterial conjugation. The insertion of the genetic material from the second organism by way of its expression in the host will confer the new desired characteristic on the GMO. So, is the question that is generally asked is genetic engineering or making a genetically modified organism different from traditional breeding? Yes, it is different from traditional breeding, although in traditional breeding also we can transfer the gene of one plant into the other, but it takes a very long time. So if you compare uh, traditional plant breeding with genetic engineering, if you look at traditional plant breeding and you have a plant, a donor plant with this gene that you want expressed in a commercial variety. So by traditional breeding, that is by crossing these two plants, you will get a progeny, and the progeny will have, in addition to the gene of your choice, it will have the other genes also being transferred. So what you get is a new plant variety, but you get your gene of choice, but also you get the other genes. Now, again, you have to cross this with a commercial variety, and it is by trial and error that you finally get the variety with the new plant. So it takes several generations by traditional plant breeding to insert the gene of your choice from one plant or from one organism into the desired organism that you want. On the other hand, genetic engineering is targeted. It is directed towards the gene that you want. So in genetic engineering, you can cut out the gene that you want and insert it into the commercial plant variety into a position that you want, allowing it to express either in the leaves or in the root or in the seed. And what you get is the improved commercial variety. So both of these techniques, that is traditional plant breeding and genetic engineering, involve the transfer of a desired trait by way of a gene into a commercial plant variety. Only A difference is this is targeted where this is not targeted, and it will take a long time. So the difference between selective breeding and GMO is selective breeding is slow, it is imprecise, and modifications of gene that naturally occur in the organism will take place. GMOs, on the other hand, are very fast, but very fast doesn't mean it's going to happen in a day or two. By the time a GMO is released, you will see in one of the slides which tells you how long it takes for a GMO to be released. It is very precise and it is targeted, and therefore it can introduce gene into any organism that would not naturally occur. So you can transfer a gene from a bacterium into a plant, from a plant into a bacterium, from a bacterium into an animal, and so on. So that is what a GMO is, and this is the difference between selective breeding and from genetic engineering. So in agricultural breeding, again over here to see, when you see what we do is selection by the breeding, and you get, it takes several generations to select and get the plant of your choice. So in genetic modification, basically what is done is you have a bacterium uh, in which a plasmid is there. You have another cell, or you isolate the DNA from another organism, be it a bacterium or a plant. You cut out that gene, you introduce it into this plasmid by molecular biology techniques. What you get is a recombinant DNA. So the small red piece is the DNA from the second organism, and then you transform the bacterium with this recombinant, and by cell division and reproduction you can select the organism containing the gene of choice. These diagrams look very simple, but however, those of you who are doing molecular biology will know that it is not this simple. So, when you talk of a genetically modified organism, the best comparison I can take and give you is, if you want to move a GPS system from your old car, which is very good, into the new car that you have bought, what would you do? Would you disassemble both the cars, mix up the parts, and then select the car which has the GPS, ignoring what else it might have picked up? Or would you just take the GPS out from your old car, just cut it out from your old car and put it into your new car? So this is almost what we have as genetic engineering. So taking the GPS from the old car and putting it into the new car. Now, the question is, and the question has been raised, is that when you do genetic engineering or genetic modification, you tend to change the other properties. So just by putting a GPS of the car, of your old car into a new car, or if you take the GPS from an aeroplane and from an airplane and put it into your car will not fly. It will only have the GPS system or the navigation system. So this is a very simple example to explain to you what is GMO, and um, you know to understand that only the property of the trait that you have transferred will be expressed, and not any other trait which will be expressed. So some definitions are there, because you hear these words: GM food, GMO, genetic engineering, LMO, and so on. So what is GM food? GM food is any food or ingredient that has been made from a genetically altered plant or animal. So a GM food is got from a GMO. A GMO is defined in that it can regenerate. So a seed is a GMO, whereas the seed powder is a GM food. The seed is a GMO or an LMO, and its powder or the lecithin that we extract from soybean will be a GM food or a GM food ingredient. Genetic alteration is defined as altering genes using the recombinant technique. Gene is a small segment of the DNA that codes for a specific protein, and our DNA is the recombinant technique, or the method, the molecular biological method we use to alter genes. So the development of a GMO in any country is a multi-step process. It goes from the lab to the field to release, which involves a large number of steps, and it can take anywhere between 10 to 12 years to commercialize a GMO, that is a GM crop. So the first step is in the laboratory, where you identify the gene, you isolate the gene, you choose the model in which you want to express it, and then you test in the laboratory whether the gene has been inserted, and then you grow the plant in the greenhouse to test for both the expression of the property as well as the safety of the plant. Now, once this is done in the laboratory, which takes about 3 to 4 years, then you need to do field trials. So in the field trials, you have two types of trials that are done. One is a confined field trial, which is in a very small area, which is cordoned off and done. And then you have large scale field trials. And here you do the testing according to the biosafety guidelines of your country. You test for the food safety, you test for feed safety, and also you test for the environmental safety aspects as per the regulations of the country. Now, once this is done, it is almost about 6 to 8 years, and then it has to be released. So to be released, a dossier has to be prepared, right, starting from the gene, sequence of the gene, sequence of the plant to which it has been inserted, sequencing of the GMO, and so on. That is all the studies, the food safety trials, the food safety experiments, the feed safety experiments, the feeding experiments, the environment experiments, etc. You make a dossier, then you submit the dossier for regulatory clearance to the country, and then it is commercialized based on the opinion of the scientific committees and the apex bodies in the country, which I will be describing towards the end of this webinar. So basically, it takes a very long time to generate a GMO and actually release the GMO. The first GMO was made in 1996, which was the flavor saver tomato, or the tomato which had slow ripening. But somehow that tomato has died, and we do not find this flavor saver tomato in the market, but we have another slow ripening tomato. So what are the tools you require for making a GMO? One of the most important tools that we require is the restriction enzymes, which are known as biological scissors. These are restriction enzymes which actually cut a gene at a particular sequence and not at any other sequence. So precisely you can cut the genes at the regions that you want. Then you need DNA. So what is DNA? You need the DNA, that is the gene of interest. So the gene is the coding system for instructions. You know that the DNA will be transcribed into RNA, and RNA will be translated into the protein. So a gene is defined as a segment of the DNA, and in genetic modification, we always talk about a gene being inserted into a new plant or a new organism. So in the central dogma of molecular biology, it is known that the gene or a piece of DNA will be transcribed into the mRNA, mRNA will be translated into a protein, and this protein will actually give the trait. It will tell you what is going to happen in the plant. So whether we have herbicide tolerance, whether we have the golden rice, or we have the slow ripening tomato, depends on the expression of the protein, which is a product of the DNA. So basically, a very simple diagram to show you the genetic engineering process. So what you do is you identify either an organism or another cell or a bacteria which has the gene that you want to take and put into B. So you do an extraction of the DNA by the conventional DNA extraction methods. And then with the pure DNA, you use the restriction enzymes to cut the region of the DNA that you want. So this small blue piece of DNA is cut with the restriction enzymes. This DNA is taken and it is transformed into the plant cell of your choice, that is the plant that you want to insert it in. The DNA will recombine with the plant cell DNA or the host DNA. Cell division will take place, and then we need to select, and you get the transgenic plant. So this is a very scientific process where the DNA is extracted, cut, and then transformed into the host cell, and then selection for the transformation takes place. So how do we put the gene into this host cells that we have? Three commonly used methods to insert the DNA, that is, to insert this small piece of DNA into the host plant cell, is done by either of these techniques. That's Agrobacterium tumefaciens mediated transformation, or by using a gene gun, which is literally a gun and you shoot the DNA into the plant, or by a technique called electroporation, where we, through giving electric current or by using some chemicals, we make small pores on the cell wall of the host cell, which allows the DNA to enter. So in Agrobacterium tumefaciens mediated transformation, so what is done is the plasmid is isolated from the agrobacterium. It is cut with the restriction enzyme A so that it's got the same ends. The DNA from the plant cell that you are interested in transforming, or bacterial cell that you are interested in transforming, you cut the cell with, you isolate the DNA, and then you cut the cell with the same restriction enzyme. So when you cut the cell with the same restriction enzyme, you have what are called sticky ends at these two. So you have the empty plasmid, you have the DNA. The DNA will join, and what you get is the TI plasmid with the gene of interest. This diagram looks very simple, but however, it is quite tedious to do an Agrobacterium tumefaciens mediated transformation. The second the TI plasmid, which goes into the bacterium, is then infected. The plant cell, that is, the plant that you want to make new, is infected with the TI plasmid. Then the transformation occurs, the DNA gets integrated, and what you get is the new gene by cell division and the transgenic plant. In the gene gun, literally the DNA that you have cut out and the gene of interest that you have got is coated with gold. It is gold particles are made of this, and then you have a gun. It is literally a gene gun where it shoots into the plant cell. So because of the impact, the holes will be made on the plant cell, and the DNA with the gold particles to give it that impact enters the plant cell. The DNA integrates with the host genome. You have a new gene, then you have cell division, and then you can select where for the transgenic plant. And the third method, as I said, is that of electroporation, where you make small holes or pores in the cell wall of the bacterium that you want to transform before putting it into the thing. So in the plant cell, you take the plant cell, you make protoplast. That means you remove the cell wall of the plant cell and you carry out an electroporation. And electroporation, as the name sounds itself, is by passing high electric current in it to it, which if you can see over here, it creates the small pores on the protoplasm, and you mix the DNA will enter, and then of course you have the new gene. And just like Agrobacterium tumefaciens mediated transformation, the plant the cells will be transformed. You do a selection and then you select for the plant. So these three methods are there to introduce the foreign DNA. I would not like to call it the foreign DNA, the DNA from the other organism into the host plant of your interest. So, what are the types of genetic modification that have been carried out by these? So, one of the major ones is to create the herbicide tolerance and insect resistance. It's not insect tolerance, it should be insect resistant. These are the common GMOs that are available, which I will be discussing in detail. Another factor is to improve the nutritional quality of the food plants, because we know that many plants do not contain all the nutrients that are required. So one of the important and authentic GMO or an innovative GMO that was made was the golden rice, which I will describe in the next few slides. So you can improve the nutritional quality. Rice, you know, does not contain beta carotene. And we have seen that beta carotene is the precursor for vitamin A. So in India, if you see India and the Southeast Asian countries, you see rice is the staple diet. So the Ingo Potricus thought that if he introduces, or you can have a rice with beta carotene and iron in it, then you could solve the problem of vitamin A deficiency and the iron deficient anemia. And so this experiment was done to make a rice containing the carotene, and you know carotene is colored, and so therefore the rice was called golden rice. For storage and transport, it is you know, you usually grow an agricultural commodity in one place and you have to transport it through long distances, and during that time there is a lot of post harvest loss. And so therefore, if ripening could be delayed, so then, you know, storage, it could be stored for at least 7 weeks and post harvest losses will not be there. Abiotic stress is trying to grow rice plants in salt water, or trying to grow rice and wheat where there is no water in the plants. Coming to the vaccines, the pharmaceutical thing, the biotechnological route has been taken to express these vaccines in plants. For example, the rabies vaccine, you know, when bitten by a dog, several injections have to be taken and it is quite painful. However, if the vaccine is expressed in a fruit like a banana or the muskmelon, it is very easy for a child to eat it. Also, you need protein in very large quantities to make a vaccine, and it is very difficult to purify protein in quantities required. However, if you can overexpress them in plants, for example, in the tobacco plant, if you can overexpress it in the seeds, a tobacco plant will grow within 3 months, you can overexpress the protein in seeds. So a large number of vaccines have been expressed in plants. Industrially, cotton has been genetically modified so that it produces long fibers instead of the short fibers, which will help in the ginning of the cotton fiber. Also, cotton fibers which are colored, that is blue, green, yellow, have been made so that you prevent the use of using these chemical dyes and causing environmental pollution. So these are some of the traits which have been genetically engineered, and I will just discuss with you at least 2 or 3 traits of genetic modification. So the first genetically modified organism was the tomato, which was known as the flavor saver tomato. This was genetically engineered so that, you know, tomatoes were produced in Mexico, and they took 7 days to reach the other end of US or Canada. By the time most of the product would be lost due to post-harvest losses due to ripening. So the need was felt to have a tomato which would have a longer shelf life, or it would take longer to ripen. And during the ripening of tomato, in the ripening process, there is an enzyme called pectin methyl esterase, which actually softens the pectin which is on the outer tube and causes ripening. Pectin methyl esterase and polygalacturonase are the 2 enzymes which are involved in the ripening of tomato. So it was felt that if these genes, that is, if the genes polygalacturonase and pectinase could be modified, then you could have a slow ripening tomato. And a technology called the antisense technology, where the gene from the same plant is taken and it is made in the reverse direction. A similar gene is synthesized and inserted in the opposite direction. And when it is inserted in the opposite direction, you know that when the DNA is transcribed, you get an entirely different protein. So, sorry, that's the same thing slide. So this that tomato with the pectinase or polygalacturonase, as I said, was the first GMO that was produced in 1996 by Monsanto. Since that was a failure, uh, a new slow ripening tomato was developed by looking at another pathway in the ripening process to see if ripening could be delayed. So you know that in most of the plants which are climacteric, that means you pluck it off the vine and then it ripens. It is the hormone. Ethylene is the ripening hormone. So we all know that fruits ripen because of the hormone ethylene, which is a gas. In the synthesis of ethylene, 2 enzymes are involved. One is the ACC synthase, that is the amino cyclopropane carboxylate synthase, and the second enzyme is the amino cyclopropane carboxylate oxidase. So these 2 enzymes are involved in the release of ethylene. So now if these 2 enzymes could be slowed down, then ethylene will not be produced. And if ethylene is not produced, then ripening will be delayed. So this was the rationale for developing a slow ripening tomato by taking the genes on ACC as well as that of the ACC oxidase and doing what is called as an antisense technology. Using an antisense technology, these 2 genes were slowed down and you have a slow ripening tomato which can be transported to faraway places and storage. So what is the antisense technology? Antisense technology is you have a gene of interest. We know that in normal conditions, the gene of interest will be transcribed into mRNA, and mRNA will be translated into the protein, and ripening takes place. So in antisense technology, the same gene is cut out and it is put back in the reverse direction. That is, if this is the 5 dash end and 3 dash end, it is put back so that this is 3 dash and 5 dash. So to give you an example, if this is the sequence of the native gene ATCGTA with TAGCAT, it is put back in backwards, so it gives you AGT. See here, AGTGCTA. So you have AGTGCTA. So it is put back into the in the reverse direction, and so when the mRNA is formed from this, there will be no translation. So therefore, the protein will not be expressed. So antisense RNA technology is used commonly to make GMOs. They are called GMOs because although the gene is taken from the same plant, it is taken reversed back, and you have to use promoters, terminators, etc., to see that it is integrated into the host genome. Now, coming to golden rice, this is a true value added fruit. It is a rice which has provitamin A enriched rice, so that if the rice is eaten, because rice is the staple diet in Southeast Asia, and so it is a means of conveying, as a mode of conveying the nutrient vitamin A, and because it contains the beta carotene or the provitamin A in it, you will see that the rice grains are yellow colored compared to the normal grains, which are white colored. So how was this done? So this came on the cover of Time magazine, and Dr. Ingo Potricus is the father of the golden rice. So what was done is, if you look at the rice, the biochemistry of the rice, it does not have any of the enzymes required for beta carotene synthesis. So therefore, you needed to put enzymes from other sources to synthesize beta carotene in the rice. So three different enzymes were required, and two were taken from the daffodil, whereas the third enzyme was taken from Erwinia. It is the gene for the enzyme was taken from Erwinia bacteria, and two genes were isolated from the flower daffodil. And all these three genes were inserted into the rice, and the rice plant expressed the beta carotene. So just let us see what were these and how it was actually done. So if you look at the rice endosperm, it contains no provitamin A. And the only precursor of this pathway, if you recollect when we talked about the plant pigments, especially of capsaicin, we had, there is the precursor, the only precursor for provitamin A is geranyl pyrophosphate. So it needed four more enzymes to synthesize the beta carotene. It required phytoene synthase, phytoene desaturase, the epsilon carotene desaturase, and lycopene cyclase to express beta carotene in it. So these enzymes were isolated from, as I said, the daffodil and Erwinia, and they were inserted into the rice genome, and the golden rice was got. So what you have to see is this geranyl pyrophosphate is there. So in the rice, only this was there. It required these four enzymes to have these four pathways to get all trans lycopene, which could be converted into beta carotene. So the first enzyme was phytoene synthase to synthesize phytoene. Then you have a phytoene desaturase to make the double bonds, then the gamma carotene isomerase to make the isomer of this, and then you have a desaturation. So all the four enzymes were engineered, and this resulted in the synthesis of beta carotene, the provitamin A, in the rice granules. So this was done for a philosophical reason, and the GMO is actually freely available. This technology has not been patented, and it said because it is for a human cause, the technology is available to anyone who wants to make the rice. So among the agronomical characteristic, the two uh, avenues, the two genetic modifications that are very popular and discussed in detail for their pros and cons are the herbicide tolerant crops and the insect resistant crops. So let us look at what are these herbicide tolerant crops. Generally, when legumes like soya bean and other plants are grown, you will see that along with the plants, there are a lot of weeds. And sometimes the weeds overtake the plant, and there is loss due to the weeds overtaking the plants. So generally, the farmers will spray herbicides. So a popular herbicide that is used to kill the weeds is the herbicide glyphosate, which has the trade name of Roundup Ready, which is used to control the weeds. But however, glyphosate not only kills the weeds, but it also kills the host plant, say maybe the soya bean or the chickpea which is grown with it. That is because this glyphosate actually interferes with the amino acid biosynthetic pathway of the normal plant. So it disrupts the function of an enzyme called enoyl pyruvate shikimate phosphate synthase, or abbreviated as ESPS. So because this enzyme, if you see, is very important in plant for the plant survival in the biosynthesis of the aromatic amino acids phenylalanine, tyrosine, and tryptophan. So when glyphosate is sprayed, glyphosate inhibits the enzyme EPSPS, and it does not allow the synthesis of this, and so therefore the plant, due to nutritional deficiency, dies. So along with the weed, the plant also dies. So this is futile, because not only do you have a loss, you also have sprayed a large amount of the herbicide. So the strategy was to try and find an enzyme, EPSPS synthase, which was tolerant to glyphosate, so that the plant could synthesize the aromatic amino acids, and the plant would grow, whereas the weeds would die. So therefore, for the glyphosate, this is the same pathway, but it shows you in structure. So shikimate is converted into enoyl pyruvyl shikimate phosphate. The enzyme is EPSPS. It is inhibited by glyphosate. So the question was how to make the herbicide tolerance. So a gene from the soil bacterium, that is the CP4 strain of Agrobacterium tumefaciens, was isolated, and it is found that the EPSPS of this strain of Agrobacterium tumefaciens was not susceptible to glyphosate. That means the enzyme was not inhibited by glyphosate. So therefore, the gene for the CP4 EPSPS from Agrobacterium tumefaciens was isolated, and it was inserted into the soya, and you have a soya bean now containing the new EPSPS, that is an enzyme which is not inhibited by glyphosate. And the Roundup Ready soya bean was born. So they have incorporated the enzyme from the soil bacterium into the soybean plant, and the soybean now expresses an EPSPS which is not inhibited by glyphosate. So glyphosate can now be sprayed and the weeds will die, whereas the soybean plants and the other plants will survive. So we have a whole range of these EPSPS plants known as Roundup Ready because it was developed by Monsanto. You have Roundup Ready soya, Roundup Ready cotton, Roundup Ready maize. So all these are herbicide tolerant. So you can spray glyphosate. Another herbicide that is commonly used to kill the weeds is a herbicide called glufosinate. This also is a herbicide which is sprayed on the plants. It kills both the weeds and the plants. So what glufosinate, which the structure is given here, also known as phosphoinothricin, is that it inhibits the enzyme glutamate synthase. This glutamate synthase is a very important enzyme in plants because it removes the ammonium ions. So glutamate reacts with ammonia and it is removed as glutamine. So if the glufosinate inhibits the glutamate synthase, then in the plant ammonia will accumulate, the amino acids will not be removed, and the plant will die because of ammonification, and the detoxifying effect is reduced. So to prevent this and to target only the weeds, uh, an enzyme which actually breaks down phosphoinothricin was isolated from a bacterium. It was inserted into the soybean plant. Now, when glufosinate is sprayed on the soybean plant, after it kills the weeds, but whereas in the plants, because of the enzyme, the glufosinate will be inactivated. So in herbicide tolerance, we have two types of plants. That is, the glyphosate is introduced, and the other is the glufosinate tolerant plants. Now, coming to the insect-resistant plants, why was there a need for the insect-resistant plants? Because if you look at the plants, most of the loss in the plants is due to the insects, especially what are called as the lepidopterans, the coleopterans, that is the worm class of thing. So this is a graph to show you the crop-wise consumption of pesticides in India, and you can see maximum pesticide usage was for cotton. So not only did it harm the environment, but it also harmed the health of the cotton farmers because it needed a large amount of the pesticide to be sprayed. And one of the pesticides that is commonly used for the cotton is the crystal proteins of the bacterium Bacillus thuringiens. So this is how the worms destroy the cotton balls. So the Helicoverpa armigera feeds on a cotton ball, and when it feeds on a cotton ball, this is the damaged cotton ball, and so you can see what type of yield loss there is in the cotton plant. So the development of the insect-resistant plants was a breakthrough, and the plants which are known as BT, standing for Bacillus thuringiens, were born. So what is BT? So if you look at BT, BT is the Bacillus thuringiens. This bacteria actually produces a toxin known as delta endotoxin. This is a toxin that this bacteria actually throws out, and it can be isolated from the soil. So this delta endotoxin is used as the pesticide. In the pesticide sprays that are used, it is the delta endotoxin which is used, and it is known to kill the Lepidoptera, the Diptera, and the Coleoptera. And the toxins which are there are toxic to a whole group of the insects, and so therefore are sprayed indiscriminately to kill these insects. So the question then arose as to what could be done to minimize the spraying of these pesticides. So actually, if you look at the taxonomy of BT, that is the Bacillus thuringiens endotoxin, it is produced by the species Bacillus, and in this subspecies of Bacillus thuringiens, it is the Kutsisaki, the HD1 strain, which produces the Cry1A enterotoxin. And you have also the other strain which produces the Cry1B, and there are some strains which also produce the Cry1F. These cry toxins, or the cry proteins as we call them, are specific for specific insect groups. So this is the crystal structure, and they have identified which part of the protein is responsible for the toxicity to the insect plants. So now the what was done to produce the GMO was to isolate the gene for which produces the toxins, that is the Cry1A or the Cry1AB or Cry1F, isolate the gene from Bacillus thuringiens, and then through genetic engineering insert it into the plant. So now the toxin is being produced continuously by the plant. So when insects come and feed on the plant, because of the deleterious effect on the insect dies, but the plant survives. So this BT, insect resistant plants, are developed for several insects, including the corn borer for the corn. So we have a BT maize. There is a BT gene Cry1AC which is put into cotton. So you have the Cry1AC cotton or the BT cotton, and BT cotton is the only genetically modified organism which is permitted to be grown in India. And since April 1st, 2002, permission has been granted. And since then, reports say that the cotton farmers have reduced the number of space, and it also has improved their yield and health. So how does the BT toxin work on the insects? So here in this case, the toxin is expressed in the plant. So the question was, if the toxin is expressed in the plant and then it is there in the seed, we eat maize, we eat soya. So is this toxic to the humans? But if you look at the biochemistry and the physiology of how BT acts on these insects, you will realize that it does not do anything in the human gut. So the gene for the BT endotoxin was introduced into the plants by genetic engineering. The gene for the BT protein becomes a part of the plant genome, and as the plant grows, the BT protein is expressed in various plants. In the case of cotton, is expressed in the cotton ball. The plant insects, while feeding on the plant, they ingest the toxin and they die. So how does this toxin work? Here is the gene construct which has there for the BT maize. So you see, it's not very simple. We cannot just put the gene in. You need to have a promoter, you need to have a terminator, you have to target it to the leaf or to the seed or to the root. But nevertheless, let us see what happens. When the insects feed on the plant, whether it is the leaf or the fruit, the protein is solubilized, and when it reaches the gut of the insect, the pH of the gut of the insect is 9.5, whereas the pH of the gut of the human body is 1.5, because hydrochloric acid is produced. So when it feeds on the plant, the Bt protein is activated in the gut because of the pH. It is actually an inactive protein. It requires an alkaline pH to be activated. Once it is activated, it binds to specific receptors which are present on the gut wall of the insect. These receptors are not present in the human gut, and once they bind because of the receptor interaction, they insert into the gut, they damage the gut wall, they cause pores to be formed in the gut, and this results in the spillage of all the gut constituents. The lysis occurs, and this results in the death of the insect. What is to be understood over here is that the activation requires pH 9.5. At pH 1.5, the Bt protein cannot be activated. And further, it requires receptors to bind to the gut, and these receptors are absent in the human. So the same thing that I had have just explained. So here is it in writing. So it injects the toxin. The protoxin gets activated in the midgut at alkaline pH. The activated toxin binds to receptors on the midgut epithelium. It creates leakage channels or pores. All the midgut cells disintegrate. The hemolymph leaks, and the insect, because the digestive system is paralyzed, the insect stops feeding and finally dies of starvation and septicemia. So common GM plants and which have been genetically engineered are corn, canola, potatoes, tomatoes, squash, soybeans, flax, cotton. So the product will be cotton seed oil. You also have sugar beets, which have been genetically modified. In fact, you have a genetically modified potato and a genetically modified apple, which do not turn brown on cutting. This is also, I think some onions have been genetically modified where the enzyme allinase has been inactivated. So you have onions where you can peel them and cut them uh, without crying. So common GM foods by trait, if you look, the maximum or the largest is that of soybeans, which is 57.5%, followed by corn, cotton, and canola. Almost all the soybeans which are grown in North America and South America are GM soybean or the Roundup Ready soybean. If you look at the nations which are growing the genetically modified, you find that India does find a place here with accounting for about 3.7% of the global GM crops. That is because cotton is a major crop in India, and only most of the cotton grown is the Bt cotton. So now comes the question of how do you identify a GM food or a GM or crop from a non-GM crop. Now, if you look at the seeds of the herbicide tolerant soya or the Roundup Ready soya, you cannot tell which is GM and which is not GM. So actually, phenotypically, we cannot tell when we look at the characteristics, they cannot be distinguished. The only way to distinguish between these two is at the molecular level, and that is by testing the protein or testing the DNA. So here is a non-GM and a GM corn. So this is the non-GM corn, and the difference in the GM corn is that it has this gene for, say, Cry1AC inserted. So this small one expanded shows you the gene of interest, where you have the promoter, the gene of interest, and it has inserted into the plant DNA. So to identify the GM from non-GM, we have to detect the presence of this new DNA, or we have to detect the presence of this the product of this DNA, which is the protein. So the introduced RNA sequence is the only one which differentiates the GM plant from its non-GM counterpart. So GMOs can be identified, as I said, based on the DNA and the RNA, because it is known that DNA is transcribed into RNA and RNA is translated into the protein. RNA techniques are quite difficult to perform, but in the biological techniques, identifying a DNA sequence or a gene or the protein is not very difficult. So the GMO analysis, you can do it either by phenotype or by the genotype. Phenotype is by doing bioassays. So in case of the insect tolerant, you can see the feeding of the insects. But however, we do what are known as the protein-based assays, and we can do it by the assay, enzyme-linked immunosorbent assay, or a lateral flow device. In the DNA-based methods, the famous PCR reaction, that is by the polymerase chain reaction, we can identify the DNA, and we either use endpoint PCR or real-time PCR. The details of the PCR reaction and ELISA and lateral flow, I will discuss with you when I talk to you about the biological techniques used in food analysis. But I will, in very brief, discuss these over here. So the protein-based assay is characterized by the antigen antibody reaction. So the proteins we know that are newly introduced are the CP4, EPS, and the Cry1A genes, or VIP3A, which is also for insect tolerance. So antibodies have been raised to these proteins. The monoclonal antibodies have been produced. And you know that an antibody antigen reaction is like a lock and key mechanism, where one key will fit the lock. And so if the antibody is tagged with a protein label which gives a color or a fluorescence, this reaction can be carried out. This is very easy to perform. It is cheap and reliable. Your tests can be done within 5 minutes. It is very good for the leaf, seeds, and grains. It is not good for GM or the GM food, that is, say, a GM biscuit or a lecithin. And it only identifies which protein is present, but it cannot tell you which event has occurred. And of course, you're able to get 99% confidence levels. So very simple test which can be done at the field. So what you do is you can just punch out a leaf or take the seed and crush the seed. The lateral strips are available. You add buffer. You can also just add water, make an aqueous extract, insert the strips into it, and then read the results. So the results that you see are, if the seed is a GM seed, so it will have the protein, it will react with the antibody, and the color will develop, or the signal will develop, and you know that a GMO is present, and you can see it by this line, that is the antigen antibody line here, whereas this is just to tell you it is the control line for the strip. Now, if the seed is a non-GMO, the protein will not be there. So whatever protein is extracted, it cannot react with the antibody, and there will be no color. And so you will not see this line of the. It is almost like the tests that are available for the pregnancy test kits, where the antibodies are used. So here are some actual experiments done with the flow strips, where you can detect even 0.01% soya, 0.1% GM soya. That means it's equivalent to 0.01 grams in 100 grams of the material. So what you see is this is the non-GM, so you have the control line, whereas in the GM you will see the protein, that is the EPSPS protein from the GM would have reacted. So it is a very simple test. You take the sample, shake it, insert the strip, and then read it after 5 minutes. However, while doing the lateral flow strips, one has to be very careful in that the strip is working. And to know if a strip is working, this line must be present. This is the test line, and that is the control line. If the control line does not appear, it is evident that the strips are not working. So here is an example which is done for the BT maize, and you can see that the test lines are visible. However, in this strip which is over here, this is invalid because we do not see the control line. So the strip cannot be used. So these are very useful, and lateral flow strips are available now for most of the proteins that have been inserted as either in the herbicide tolerant or in the insect resistant plants. Then we have the DNA-based testing, which is the more accurate form of testing, and this requires identifying this small piece of foreign DNA which has been inserted. So how is this DNA detected? DNA is extracted from the seed, and then this small portion of the DNA is amplified by the polymerase chain reaction, which amplifies or converts the minute amount of DNA into larger amounts, which can be visualized by gel electrophoresis or by real-time PCR. So the polymerase chain reaction detects a known recombinant DNA sequence. The target sequence is duplicated billions of times using a thermocycler and the Taq polymerase, and results obtained can be either quantitative or qualitative. So what is required for a PCR reaction is the DNA that you have isolated. You need the forward and the reverse primer. You need the Taq polymerase, and you need the nucleotides. And the basis of the basics of the polymerase chain reaction will be discussed in detail in a webinar in the future. So in the PCR reaction, you can think of the PCR reaction as though it is a photocopying machine. So all of you, if these PowerPoints were available, you would, each one of you would photocopy it, give it to a friend, that person would photocopy it. And so you can get millions of copies by just this wonderful machine which does the photocopying. The PCR reaction or the PCR machine can be compared with a photocopying machine, except that in a photocopying machine, you can get only one copy at a time, but in a PCR, the DNA will be copied 2 times in every cycle. So in the first cycle, if you have 2 copies of DNA, in the second cycle, you will have 4 copies. In the 10th cycle, you will have 1024, that is 2 to the power of 10. After 30 cycles, that is what we generally do, what you see is we have more than 1 million copies, and the DNA is easily visible. That is the power of the enzyme polymerase. the DNA polymerase and the polymerase chain reaction. So if you want to do it from oil, if you want to detect whether the oil has been extracted from GM soybean or from non-GM soybean, you could only do it from crude oil, because crude oil may give you some DNA, refined oil does not give you. So it involves extraction of the DNA, getting the purity of the DNA, synthesizing the proper primers. So you have a forward primer and a reverse primer, amplifying this by the polymerase chain reaction, and then detecting it by gel electrophoresis. So if this is the foreign gene of it, if you see that the gene of that thing sequence, then you know that it is a GMO. And in PCR, we do what is known as endpoint. That means I'm looking at the DNA at the end of 30 or 35 cycles. And this tends to be a little bit on the saturation side. And it is very good for the qualitative analysis. And if you look at this figure, it shows even if you have 10 copies of the gene and 50 copies of the gene, you cannot differentiate between the two. So if you want to quantitate, you cannot use the endpoint PCR, and we need to use what is known as the quantitative PCR or the qPCR. And this qPCR is an instrumental method. It is a polymerase chain reaction, but is carried out in an instrument which actually measures the amplification in the linear part of the reaction and not in the plateau. In the previous gel, what we do is we look at the DNA at this region. So it does not differentiate between 0.1% and 1%. But whereas if you do it using the quantitative PCR, you will be able to tell the exact quantity of the DNA that is present in the sample. So the endpoint PCR has poor precision, lower sensitivity. It has a very short dynamic range. It's only a sized brace description, and results are not expressed as numbers. Also, those of you who have done gel electrophoresis and PCR will know that you use ethidium bromide, which is a carcinogen, and one has to avoid using ethidium bromide. So to overcome this, we use a reaction called the quantitative polymerase chain reaction, in which, in addition to the primers, we use a third probe, which is a fluorescent probe. And this fluorescent probe, as the amplification proceeds, will give out fluorescence, and the fluorescence that is measured is proportional to the DNA there. And as I said, the details of this reaction, I didn't want it to be a repetition, so will be done in the webinar when we do biological DNA and protein-based methods in food analysis. So this is how the PCR amplification will look. So as you can clearly see, when you have less of the GMO, it takes a very long time to amplify. If you have more of the GMO, it amplifies very fast. So by just looking at these cycle numbers and plotting a graph of the cycle numbers versus the concentration, we can tell the amount of the DNA. So here is a graph, and these are graphs which were produced in my laboratory when we were doing GMO analysis. So here what you can see in these colors are the standard GMO or the reference GMO. So this is 10% and this is 0%. So a graph is plotted of the CT value, that is the cycle number, which is over here. So the CT value versus the GMO concentration. So what you get is a straight line, and what is shown over here in red is the sample. So if you look at the sample there, by calculation we get 0.72. Now, if you look at this graph, this 0.72 is very close to this cyan graph, which is about 1.6. So therefore, it is quite close. And I will, as I said, in the class on biological techniques, the exact methods will be described. Now, we know how to analyze on thing, but how do you release it into the market or into the field? In India, the regulation of GMOs or LMOs or to release it into the field is governed by rules 1989 of the Ministry, the Environmental Protection Act. So these rules cover the manufacture, use, import, export, and storage of hazardous microorganisms and genetically engineered organisms. So just like we have the Food Safety and Standards Authority of India, the GMOs, that is the release or the growing of GMOs and LMOs, is governed by the Environmental Protection Act. So all this includes field trials, manufacturing. Even if you want to import the seeds, you have to take permission from the Ministry of Environment and Forests. So the regulation covers what is shown here. It covers all aspects of the GMOs, starting from the experimental stage. So to do any recombinant DNA experiments, you as a student or thing cannot just straight away start doing it. You have to have permission from your Institute Biosafety Committee, which is constituted under the Department of Biotechnology. And then if you want to do greenhouse trials, you have to take permission from another committee. And then if you want to do confined field trials, you have to take a permission from the Ministry of Environment, and final release also Ministry of Environment and Forests will give you the permission. So making a GMO, release of GMO are all regulated, not only in our country, but in other countries also. So the implementation mechanism for this are, we have an advisory committee, which is called the Research Advisory Committee. We have the approvals, which your initial experiments have to be approved by your institute's biosafety committee. Then you have to get approval for greenhouse trials from the Research Council on Genetic Manipulation under DBT. To do the confined field trials and large-scale field trials, you have to take approval from the apex body, Genetic Engineering Appraisal Committee of the Ministry of Environment, Forests, and Climate Change. And release also is approved by GEAC, but it is the government which has to give the order for release. The enforcement, to see whether you're following the rules, especially when you're doing confined field trials or large-scale field trials, you have the district level committees and the state biosafety committees. So these two committees will monitor the field trials and all the data that you produce for GMO. For you to understand and know all the rules and regulations, all these manuals and guidelines have been published by the Ministry of Environment and Forests and DBT, as well as Indian Council of Medical Research. So you have the recombinant DNA safety guidelines, you have revised guidelines for research in transgenic plants. We also have the biosafety guidelines by ICMR. If it has to be used as a food, what are the tests that have to be done? How do you prepare a dossier? How do you submit it? So protocols are given, how to do acute oral safety studies in rats and mice, how to do the subchronic feeding trials in rodents, to do the protein thermal stability assays, pepsin digestibility assay, livestock feeding assays, because if these GMOs are going to be used for livestock, they also tell you how to do the assays for substantial equivalence, as well as the safety of the food. So these guidelines are all available free of cost, easily downloadable from the websites. Now, coming to the food, if you look at the Food Safety and Standards Act, under Section 22, we have the genetically modified foods, organic foods, functional foods, and proprietary foods. In the Act, it says that no person shall manufacture, distribute, sell, or import any novel food or genetically modified articles of food, irradiated food, organic food, of course, all this. And the definition for genetically engineered or modified food is given as food and feed ingredients composed of or containing genetically modified or genetically engineered organisms obtained through modern biotechnology, of food and food ingredients produced from but not containing the GMO. So in this context, the regulations from the GMO are in the pipeline. They are being framed by the committee. We have a panel on GM. We have a scientific panel on GM, which takes care of this. And this, once they have made the guidelines, it will go to the scientific committee. The scientific committee will approve of this, then it goes to the Food Safety and Standards Authority. Currently, till these regulations are in place, the Ministry of Environment and Forests was clearing this. The guidelines on labeling also, the draft guidelines have been published. They are draft and open for comments, where it says that any food containing GM above 1% must on the label carry contains GMO. So the GM food approval is approval of GM foods is by FSSAI, would be in respect of the food safety, but any aspects relating to their environmental impacts will be by the GEAC. So this covers the broad aspects of GMO, what GMOs are, how GMOs are, why they were made, what are the major genetic modifications, how they are tested, and the regulations in our country for the making of GMOs and for the release of GMOs. So the next webinar is tomorrow, which will be an introduction to nutrition.